Today's topic includes receptors and process of myelination. So receptors, these are the specialized sensory nerve endings, which are classified on the basis of their function and anatomical structure. So according to function, we have mechanoreceptors, thermoreceptors, nociceptors, electromagnetic, and chemoreceptors. Anatomically, we classify the receptors into non-encapsulated and encapsulated. The non-encapsulated are further divided into free nerve endings, Merkel's disc, and hair follicle. Free nerve endings, they are present throughout the body, and their function is to perceive pain, crude touch, and temperature, particularly heat. And they are those receptors which adapt very fast. Merkel's disc are located and they can be found within the hairless skin. They are basically mechanoreceptors and they also can perceive the crude touch. They are slow to adapt and they comprise of A-beta fibers. Hair follicle, the name is indicating their location. They can be found within the hairy skin. Again, they are mechanoreceptors, perceive good touch, adaptable very fast, and they contain A-beta fibers. The encapsulated type of receptors include tactile corpuscles of Meissner's, Pacinian corpuscles, Ruffini's corpuscles, and bulb of Cross, muscle spindle organs, and Golgi tendon organs. Tactile corpuscles of the Meissner's, they can be found within the hairless skin and external genitalia. And they may extend up till the dermal papillae. They carry the sensation for fine touch and two-point discrimination as well as pressure. And they are fast to adapt and contain A-beta fibers. Whereas Pacinian corpuscles can be found within the dermis of the skin, ligaments, joints, as well as external genitalia. They are onion-shaped, hence their name is Pacinian. They are mechanoreceptors, perceive vibration and pressure, fast to adapt, and contain A-beta fibers. Ruffini's corpuscles are found in the dermis of the hairy skin. And they're again mechanoreceptors, respond to touch, temperature, especially warm, heat, slow to adapt, and are A-beta fibers. And bulb of Cross found in the dermis and perceives the sensation of cold. Muscle spindle organs, the name is indicating they can be found within the skeletal muscles, mechanoreceptors, so they respond to stretch, especially lengthwise. They're fast to adapt, have A-alpha fibers and beta fibers. Golgi tendon organs, again, the name is explaining their location. They can be found within the tendons of the muscles. Mechanoreceptors, so they perceive stretch, especially tension related, fast to adapt and have A-alpha fibers. This is a diagram showing different receptors. Here we have onion-shaped, having various layers like an onion, Pacinian corpuscles. We have Ruffini's, Merkel's, Meissner's, Cross, and bulb. These deeper, as you can see, most of them can be found within the dermis. A few can extend up till the dermal papillae like Meissner's and Merkel's. Moving on to the next topic, which is the nerve fibers. We have seen in the previous lectures that nerve fibers refer to axons and dendrites, the fibers basically arising from either side of the cell body, neuronal cell body. These are the bundles of the nerve fibers in the CNS. So the bundle of nerve fibers located within the CNS are called tracts, whereas similar bundles of nerve fibers, when found in the peripheral nervous system, they are referred to as peripheral nerves. Classification of nerve fibers. We can classify the nerve fibers on the basis of two main criteria: by the speed of the conduction and size, and according to degree of myelination. According to degree of myelination, the fibers can be classified as myelinated and non-myelinated. And by speed of conduction and size, we divide the nerve fibers into A, B, and C fibers. All of these fibers have different conduction velocity, diameter, function, degree of myelination, and sensitivity to local anesthetic. A fibers, they are further classified into alpha, 1A, 1B, beta, 2, gamma, delta, 3, and here is their speed of conduction with varying degrees of the diameter. Their function is mostly sensory and sometimes motor. Most of them are myelinated and they're supposed to be least sensitive to local anesthetic. The type B fibers, they have 13 to 15 meter per second conduction velocity. Diameter ranges up to 3 micrometer, can be found as preganglionic autonomic fibers, and they are myelinated. Type C fibers, also called type 4 fibers, they carry diffuse pain, temperature sensation, can be found within the postganglionic autonomic fibers. They are non-myelinated, hence they are most sensitive to local anesthetic. So the process of myelination is a bit different in both types of nervous systems, that is peripheral and central nervous systems. In general, the process of myelination begins before birth and first year of postnatal life. In peripheral nervous system, the process of myelination begins with the indentation of the side of Schwann cell by the neuron. So the indentation that is produced within the plasma membrane of the Schwann cell forms a mesexome, which suspends the neuron as it sinks in the Schwann cell. Here you can see that on the right side of the slide, this is a Schwann cell, and within its plasma membrane, an indentation has been produced by the neuron. This indentation is labeled as mesexome. Further in the process of myelination, Schwann cell wraps around the neuron in spiral fashion. Initially, the wrappings are loose, but they become tight with disappearance of the cytoplasm. And the thickness of these spirals depend on the number of spirals. So that this lamellated appearance is about 13 to 18 nanometer in thickness. During the process of myelination in the peripheral nervous system, two types of thick and light bands can be seen. The major dark dense line is produced as a result of the fusion of inner cytoplasmic protein layers of the plasma membrane of the Schwann cell. Here you can see the dark dense lines, which are also called period lines. The outer or external plasma membrane of the Schwann cell that mostly comprises of the lipid fuses to form a minor dense line, which is also called intra-period line. At node of Ranvier, the Schwann cell terminates and myelin becomes thin. Here you can see a node of Renvier where the myelination around the neuron before this node of Renvier comes to terminate at this level, and so that the exolemma is exposed. Incisors of Schmidt-Lenterman, they can be seen on longitudinal cut section of the neuron. In the longitudinal cut section of the neuron, dark major lines cannot be formed. Localized persistence of the cytoplasm of Schwann cell can be seen, involving all layers of the myelin. And in the area of the incisors of Schmidt-Lenterman, since dark major lines are not formed, it provides conduction of the pathway of metabolites from surface region to the axon. So this is the significance of the incisors of Schmidt-Lenterman. Now, some differences in the process of myelination taking place within the peripheral nervous system and central nervous system. We know that Schwann cell is responsible for the myelination in the peripheral nervous system, but in the central nervous system, it is the oligodendrocyte that leads the process of myelination. In the peripheral nervous system, we just saw that axon was formed, which was basically an indentation within the plasma membrane of the Schwann cell. But no such structure is formed in the central nervous system because it is the oligodendrocyte that extends its process to wrap around the neuron, instead of the neuron producing an indentation within the plasma membrane of the oligodendrocytes, as happens in the peripheral nervous system. Secondly, nodes of Renvier are definitely present, and so are the incisors of Schmidt-Lenterman. Composition of myelin is that basically it comprises of primarily the lipids, water, and protein. And the most commonly found lipids in the myelin include cholesterol, phospholipids, and glycosphingolipids. Function of the myelin sheath is that it helps to increase the conduction velocity of the impulse along the neuron. It reduces the energy that is spent in the process of conduction, and as well as it is this myelin sheath that is responsible for the color of the white matter within the brain and the spinal